Hello and welcome to the Microbiomedics podcast. I'm Dr. Siobhan McCormack.
And I'm Dr. Sheena Fraser, and we're your co-hosts.
We are both GPs and lifestyle medics with a shared passion for microbiome science. And we have spent the last 5 years deep diving into the world of the gut microbiome. We'll be translating the evidence and packaging it into actionable, bite-sized chunks so that you can harness the power of the microbiome to improve your own health and that of your patients.
Just before we start this interview, we'd like to remind our listeners to take a look at the Gut Microbiome for Clinicians course. It's available through bslm.org.uk. It's a comprehensive course on the gut microbiome which takes you from the basics to the clinical application of this fascinating new area of medicine. This course will empower you to use microbiome science in your daily practice. Hello and welcome to another episode of the Microbiomedics podcast. I've got Siobhan McCormack with me. I'm Sheena Fraser. We're both busy GPs and we are both obsessed with everything microbiome. So today we're going to do something slightly different. All up until now, we've really been focused on the gut microbiome, which is, of course, the biggest human microbiome we have, the most in-depth and the most studied human microbiome. But I think, it's important to realize that this body of ours houses lots of different microbiomes, and actually some of them are fantastically interesting and are just as important for human health as the gut microbiome. So what we're going to do is this episode is going to be focusing on the vaginal microbiome, which is one that I think a lot of people might not automatically think of as being an important microbiome in the human body, but we do. And Siobhan has interviewed somebody who is a scientist and an expert on the vaginal microbiome. So Shiv, tell us all about your interview and tell us why we decided to do this.
I got to speak to the super busy Professor Sarah LeBeer, who is an amazing microbiome scientist who's really dedicated her whole working life to this area. So she is a professor at the University of Antwerp in Belgium. She runs the LeBeer Lab, and she's got a group of people, think about between 20 and 30, that study beneficial microbes. And particularly her passion and obsession is lactobacillus and lactobacilli. And she looks at the genus of microbes and their ecological context. She looks at the evolutionary history, and she's really trying to deep study the core mechanisms behind the beneficial potential of this incredible group of microbes. I was just so fascinated by what she was saying. She was so interesting and she's so knowledgeable. I could have spoken to her for another 2 hours. But yeah, what were your thoughts?
Yeah, well, we're only doing one episode dedicated to the vaginal microbiome, so we want to make it a good one. And I think that this is a really interesting project, and there's a lot of very important information that comes out in this. You are going to get a lot of very, very interesting information on the vaginal microbiome. And, I think, have a listen to this. But what I'm interested in is taking this knowledge forward and thinking about how we can use this knowledge. So as a GP, for me, I want to know the practicalities. I want to know, okay, we have this knowledge about vaginal microbiomes and what kind of species are in there, and this is what you're going to find out in this interview. But how do we then apply that knowledge and relate that to the sort of diseases that we see in the vaginal microbiome and throughout different times of our life? And, that was a really interesting thing about this, that, it's really important to remember that the vaginal microbiome does change throughout a woman's life. It's very, very different pre-pubertal. It becomes different again in sort of early puberty. It changes in middle years and with pregnancies, and it changes again postmenopausally. So at different stages in a woman's life, they're going to have different issues with the vagina, and that can make them more prone to different diseases. So it's really interesting. I think as a GP, I see a lot of women with issues and diseases of the vagina, and it's important to know how these come about and how we can prevent them.
I think it's probably helpful for us to just discuss the most obvious differences between the gut microbiome and the vaginal microbiome. So the listeners on this podcast will be aware that the gut microbiome, we're always talking about a more health promoting ecosystem, is one that has increased diversity, richness and abundance. So we want lots of different types of microorganisms. The vaginal microbiome is strikingly different in that they're looking for a reduced diversity. So a more health promoting vaginal microbiome contains mainly this genus, Lactobacillus, which is a very complex genus and has many, many different species and subspecies in it. But they describe 5 different types, I suppose, that are mainly dominated by one or other of these lactobacilli. So the 4 main ones in the vaginal microbiome are Lactobacillus crispatus, Lactobacillus inners, Lactobacillus jensenii, and Lactobacillus gasseri. Do not worry about the names. I'm just showing you that there are 4 different types. And there's a 5th type, which is a more pathogenic community state type, which is kind of a mixture of pathogens, anaerobes, things like Gardnerella and Prevotella. Again, don't worry about the names. Just know that there is this less, I suppose it's more, it would be the equivalent to a state of dysbiosis. And this is the state that's associated with bacterial vaginosis and other vaginal disorders. I'm going to hand you over to the expert. I mean, one of the world's experts in this area, the wonderful Professor Sarah LeBeer, who I'll start off by just asking her how she became interested in this area. Here we go.
Yes, actually. So I have a master in bioscience engineering. I think it doesn't exist in every country, but it's in a typical Belgian degree where you want to explore all aspects of the human body, the animal body, plant science, and it's more biotechnology centered. And I got into the field of the microbiome when I did my master thesis, so I could choose a topic. And I had a professor who was working on pathogens, but also on this new field of beneficial microbes and probiotics. And I was really fascinated by the beneficial aspects of microbiology. So I actually started in the field by studying the mode of action of a well-known probiotic and how it could improve gut health and reduce gut inflammation. So that was my PhD research. And then I met another professor working on HIV, and he was fascinated by I wanted to explore whether the normal bacteria in the vagina could help women protect against HIV infections. So that was my first introduction to the vaginal microbiome field. And then a few years later, I had the opportunity to start my own research group. And as also typical for this podcast, as so many people are already working on the gut microbiome. So for my own research team, I thought, okay, let's focus on microbiomes of the human body where there's less research. So we decided to focus on the vaginal microbiome and also the upper respiratory tract microbiome and also a little bit on the skin microbiome. And often I'm really fascinated by the daily light aspect of the microbiome. So I collaborate a lot with GPs, and because I think a lot of diseases or disorders or discomforts are linked to the microbiome and we don't know a lot about it yet.
Yeah, that's it's really interesting what you say about the gut microbiome, which is what the podcast has focused on to date. And you'll be pleased to know that the first area we're going to explore outside the gut is the vaginal microbiome. So it's incredibly exciting to be with you talking about this today. And I think it's one of those subjects in society that's either, when people talk about vagina, they talk about it's either sexualized or medicalized or sanitized. And it's this wonderful opportunity to reframe our conversation in ecological terms, the idea that we've all got these incredible microbiomes, the gut is the best known. So the podcast has talked a lot about the gut microbiome. Can you introduce us to this metropolis, this microbiome in the vagina? Can you tell us something about it?
So there's one thing, and that is linked to the gut microbiome, because anatomically there's a close interaction between the gut through the anus region and the vagina. But it's a totally different environment. It's what I'm a microbiologist, so I like to see it as like a bioreactor. It's a whole collection of microbes we females have in our body. It's a very highly concentrated. Sometimes even the density is as high as in the gut, but it's a much in ecological terms, it's a much simpler community. So normally when women are healthy, they have only a few species that are dominating or really living in the vagina. And the typical species are lactic acid bacteria or lactobacilli. And they have a main function, and that is producing lactic acid. And that lactic acid, the main function we always think is that it keeps pathogens away. So it makes sure that our crucial organ or our crucial reproductive tract is protected against pathogens, either bacteria, yeast, or fungi, and even viruses. But with my team, we also always say that this view on the vaginal microbiology is really too simple. They will do much more than just producing lactic acid and keeping pathogens away. It's like a whole metabolic organ with a lot of extra functions we don't know much about.
So just going back to what you said, because when we talk about the gut microbiome and we're trying to work out how we can describe a more health-promoting gut microbiome, one of the things is increased diversity and richness and abundance. So what you're saying about the vaginal microbiome is that it's more health-promoting if it's less diverse, if it's just lactobacilli. Would that be correct? Or how is it? What's the framework you use?
Yeah, I think in the framework is indeed the opposite. So the less diverse, the better. It depends a little bit on what are then the dominant bacteria. So we know if there are more lactic acid bacteria, and if they're the dominant ones, so the community is less diverse and it's associated with more healthy outcomes. So less problems with pregnancy, less problems with inflammation or redness or unpleasant odor. So the less diverse seems to be the dogma, although we are not sure yet. It also seems to depend a little bit on where people live, and there also seems to be some hidden diversity underneath. So I think the idea is not that you just need one microbe in your vagina. There's still a community of different microbes collaborating together.
Yeah, which brings me on to lactobacilli, because when you're new to this area, it's very easy to think bifidobacteria is, one type and lactobacillus is another type. But this is this very complex and heterogeneous group of bacteria. Can you tell us a little bit more about that? Because it can sound simplistic to say it's just lactobacilli. Can you introduce us a little bit to this very complex family of bacteria?
Yeah, so in the vagina, the genus Lactobacillus is important. It's one of the most ancient groups of bacteria, and there are actually 4 species that are commonly found, and the most important one is Lactobacillus crispatus. For example, in our own Belgian cohort, we found that around 43% of the women are dominated by Lactobacillus crispatus. Then you have a second species, which is also important, is Lactobacillus iners. It's also very prevalent, so around, I think, more than 20% of the Belgian women have a Lactobacillus iners dominated microbial community. And then we have Lactobacillus jenseni, which is number 3. And we have number 4 is Lactobacillus gasseri. And these are the most common Lactobacillus species in the vagina of women typically living in Western areas. So also, I think in the UK, it will be the same as in Belgium. So there are different species and they have a lot of common properties. So they all produce lactic acid, but they also have a lot of differences and different metabolic capacities, they're a little bit different mini factories producing other molecules. And actually, we don't understand why only these 4 species are so common. And we also don't understand why one woman has a crispatus dominant community and the other woman has an iners. And we also don't know whether they can also co-occur. So sometimes you just have a mixture, and sometimes you switch after menstruation. For example, one woman can become iners dominated, and the next cycle she has crispatus. So there's a lot happening in the vagina that we still need to explore.
I think a lot of people still struggle with the idea of, microbes being beneficial. We're in a world, certainly with my medical training, we only focused on pathogens or these microbes that caused or could potentially cause disease, and vaccinations and antibiotics. And of course, with the COVID-19, that's pushed people even more towards this idea that all microbes and viruses are negative to health. But what you're saying here is that we have this community of microbes living in the vagina that produce lactic acid, which presumably drops your pH, makes it more acidic, which is deters potential pathogens. Would that be correct?
Yes, and indeed, that's what I really love about studying lactobacilli, because I'm studying them for more than 20 years, is the fact that they're real core beneficial bacteria. So they don't have any virulence properties. They lack all the potential toxins. They're actually also producing molecules that improve your immune system, that reduce inflammation. They're in a way, very soft bacteria. They also promote even good interaction with your mucosa. Maybe the only little disadvantage, sometimes they produce a little bit too much acid, and then can also be a disadvantage. But they're really good examples of beneficial bacteria.
So when I focused there with my question on just the pH, that's far too simplistic. That's just one of the many things that they do. So we're talking again in a similar way to the gut microbiome, that you're getting conversations or crosstalk between metabolites produced by these vaginal microbes and presumably the epithelium and even more distant organs. Would that be correct? Is there that sort of thing going on?
That's definitely correct. And that's also the fascinating thing. I think the crosstalk between the bacteria and the immune system, it starts to become explored, and we start to better understand what they produce and how it can interact with certain immune receptors. The crosstalk with other microbes, that has long been ignored because they thought, okay, it's a simple community, they can live on their own. And for example, at this paper we recently published in Nature Microbiology, one of the most novel findings novel things we discovered was that they collaborate, and we see networks of even lactobacilli, different species co-occurring. So they seem to need each other for food. They exchange nutrients. And then another fascinating thing we recently discovered in the lab is that some vaginal lactobacilli, they produce vitamins like vitamin B2 or riboflavin and even folic acid, which is also important for women's health. And there's something really fascinating. And now we are thinking, why would they produce it? To promote the health of the woman in which they live? Or is it also because they also need vitamins themselves and they need it to grow and they exchange it as a nutrient? So, but it was, for example, a metabolite I never thought they would produce, but by serendipity we found it in the lab. And another area I'm really fascinated, but we haven't done real work on it. We just know that some genes seem to be present in the genome, is that they seem to be able to produce neurotransmitters, or at least neuroendocrine metabolites. And I really would love to explore whether if they produce it, whether it can go to the bloodstream and even have systemic effects, because you were mentioning John Krine's work and the psychobiotics, and we only think about them being present in the gut, and I would love to explore whether a slimmer mechanism is happening in the vagina.
Wow, Sarah, that is absolutely fascinating. And it's just ringing so many bells from, looking at the gut microbiome. And I suppose Sheena and I, when we talk about it, it's really this incredible communication between you and your indigenous microbes, and that they talk through these different channels, if you like. So we talk about different languages, we talk about immune languages, we talk about hormonal languages, and it's this wonderful idea that they're producing all these metabolites that are communicating with other tissues and organs in the body. And just going back to the B vitamins, because I think in the gut, I'm right in saying that the gut microbiome or microbiota can produce all 9 B vitamins. So you're saying it could be similar in the vagina as well?
Yeah, at least we know that some species, they produce these vitamins. So we know, I'm a molecular genetic, so we see in the genomes that they have the capacity to produce it. We see it in the lab. We see it when we isolate them from the vagina. Whether it's really happening in the vagina and has a function, we don't know yet. But thinking about what is happening in the gut microbiome research, I think it must be there, or it must be possible.
Oh, it's really exciting. What I think we should do, Sarah, then is go, because you've mentioned this paper, which is fantastic. I've got it here. I know I'm not meant to print things out, and I'm meant to be preserving paper and trees, but I'm a bit old school, and when I get a nice paper, I like to feel it and scribble on it and look at it. So I printed it out.
I fully understand. I have the same.
Yeah. So if I can say, so it's called a Citizen Science-enabled catalogue of the vaginal microbiome and associated factors. It's published in a very prestigious publication, Nature Microbiology. It was published in October 2023. We will put links. We really encourage people to have a look at these papers. We quite often, at each podcast, we usually introduce 1 or 2 papers. And even if there's people listening out there who, don't have a science background, these papers are so beautifully sort of produced that actually, if you just look at the abstract, the introduction, you can learn so much. And this one is really lovely because it's a collaboration between you and your very clever scientists in your laboratory with citizens. And so tell us a bit about it. And what it's named after and why you started it.
Yeah, the reason I started this was I already told you that I was studying HIV and infections and the microbiome and how the vaginal lactobacilli could inhibit pathogens. But at a certain moment, I thought, actually, we don't know well what's happening under normal conditions in all women and under healthy conditions. So I wanted to take a step back and study the biology of the lactobacilli in healthy women. And typically, and I think it's the same in gut microbiome research, but also in immunology, what people do is it's using mice or animal models to explore fundamental questions. But one thing you need to know is for the vagina, in mice, there are no lactobacilli. So the vaginal microbiome is totally different. And also there's actually no animal where you see this dominance of lactobacilli. So it's really something typical for humans. And it's something that really fascinates me. Why is that the case? So I thought, I want to study the dominance of lactobacilli, why humans have this unique association. And I want to study it in healthy conditions. The only way to study them properly is by having human volunteers willing to donate samples. Now, these are very intimate samples. I've done some other microbiology projects in the past, and I was just pitching to my friends. So I have this new idea. I really want to study it. I want to study it outside the hospital. Without gynecologists, but just self-sampling. Would you be willing to participate? And then I remember they said, Sarah, it's a nice idea and I think it's important, but you'll have to embed it in a little bit in a nicer sampling campaign. You have to think about communication. So we thought, indeed, if we are going to ask women to donate samples, we have to give them back something in return. It's a unique opportunity to talk about the microbiome in general, but also the vaginal microbiome, the vagina in general, and it's a part of the human body. Many women don't know all the details about. It's linked to so many things: the menstrual cycle, hormones, the way we feel, our sexual activity. And I wanted to do it in an honest way. So I decided to spend time on the recruitment campaign. I also thought it would be quite difficult, so I wanted to have a good interaction with communication specialists, sociologists, GPs, very important. And they told us that many women that come to their practice, they have problems with their vaginal health, and often they don't really dare to ask, or it's part of another question. So we spend a lot of time in interacting with them and asking them, what do you think we should study? My interest is the microbiome, but what's your interest? Also, we talked a lot with how do you say it, sexual education, non-profit organizations we have here in Belgium. And then in the end, we decided, okay, let's go for a microbiome sampling campaign, but on one hand, but it in a bi-directional way, so that we have also things we can do in return. One of the first things we had time to do was decide on a logo and on a name. So we named the project after Isala van Dist. Isala is the first female doctor in Belgium. She studied more than 100 years ago. Actually, she was not allowed to study medicine in Belgium. She had to go to Switzerland. As she came back with her degree, she wanted to open a practice. She was not allowed because she was a woman. They had to really change the law, and then she was allowed to open a practice in Brussels. And from the beginning, she was really focusing on women's health and vaginal health because, for example, she also had a lot of prostitutes among her patients. So it was also really to honor her. And also the logo of the project, for example, it's like an underwear logo with some nice pink and petrol blue colors. And for me, the key thing was that it was a very scientific project, but not only that. So we wanted to have a lot of humor and a nice tone of voice, and we wanted to also have a tote bag and some like a nice sampling box, not too medical, but also to honor the woman participating. And then we had interaction. For example, we had conversation starters, and we asked women, what should we explore? What should we add to the questions we will ask? What do you think is important? And so that was a start of a whole, what we call a citizen science project and then co-creational projects.
Oh, that sounds wonderful. And when I was reading through the paper, I think, introducing all these different areas and all these areas of expertise and citizens themselves. I mean, how did you find, what was the take up and what was the response?
So we did all the efforts because we wanted to recruit 200 women, because at that moment we had a look in the literature. I think most studies were with up to 100 individuals. And even though we knew that the vaginal microbiome is not as diverse as the gut microbiome, we really wanted to explore diversity in a proper way. And so you need enough samples. So we thought, okay, with 200, we would already be one of the best studies in the world. So we did all the efforts for the 200 participants. Everybody told us it was going to be very difficult. They all warned us, it will be difficult to recruit them and you will have to pay a lot of attention. So then we were ready to launch a call, and we chose International Women's Day, so the 8th of March. But then the pandemic started, so we decided to postpone. But then at the end of March, I thought, we spent so much effort and everybody's telling us that it will be difficult to start this project. So we thought, okay, let's just launch a call anyway with just a small press release, and we will use Twitter and our Instagram with the younger PhD students and my generation and older generation. But the response was overwhelming. I think within 10 days, we had more than 6,000 women willing to participate. There was no shame at all. The whole families were willing to participate. There was so much enthusiasm. Many women said, finally, it's important to explore and we will study that. So we did that.
Wow, that's, it's an incredible story. Because I'm just thinking when the NHS, when we try and make people come in for tests or tests for sexually transmitted disease, you're constantly trying to get people in. I think we frighten them away. It's very medicalized. What happened here that because I read that within 10 days you had over 6,000 people registering and you actually had to close it down. What's going on here? Where's this energy and enthusiasm and wanting to belong to, something like this? What's happening? What's your thoughts on it?
I think that was happening. Some people said, it's part of what women want to take health into their own hands, and it should not be medicalized too much. So that was also important because we had very good gynecologists, university gynecologists participating, but we said, no, we want to take it outside the hospital. It's not medical. We will study the normal function. It's not linked to disease. We want to also highlight that a lot of things are probably normal, so that we wanted to scan the whole health spectrum, not alarm people. And many women felt a sense of belonging. So the nice thing is we didn't prepare for it, but a lot of mothers with their daughters participate. We had grandmothers and then the grandchild participating. We had people in the elderly home willing to participate and a sense of belonging. Okay, and many women, we did a feedback survey afterwards. They said, they wanted to do something in return to other women. So some said, I have 3 healthy children. I want to help other women in the future because my little data point it will be part of a whole collection, but I can help the future generation of women with certain solutions we are not even aware of. And other women, they gave us advice. It might be nice. Maybe you can also add a question on the type of underwear we wear, because I think it's important. Other women said, you also should also ask about food intake. And we collected all suggestions, and then we added some to the to the questionnaire. So I think it was also said like we made 10 amendments to the ethical committee file because we had so many tips from the citizens to include.
So, Sarah, what you're saying is when you're calling these women citizen scientists, they truly are contributing in so many ways to the science and into how science is done. I suppose I find this incredibly interesting because I looked at the age range from 18 to 98, you had samples.
Yes.
And it's just wonderful, this, all these generations joining in, wanting to learn more and contribute. And I think for me, reading through this, it was a real reintroduction of the vagina into society as, as something other than what we're used to. There's a lot of stuff about people being concerned. People don't understand the vagina, what it looks like. Young women now aren't really taught about it. They don't know what's normal. They don't understand physiological discharge, or that slight odors can be absolutely fine. And there's a huge amount of money spent on products when this is essentially a self-cleaning organ. And people are made to fear this area. So, as I said, it's either sexualized or medicalized by people like me when we ask people to attend for their smears or to get tested. And actually, it's this kind of beautiful organ that is, doing so much good and has this community of microbes that are protecting not only the women, but their partners, their offspring in so many ways. And it's just a lovely new perspective.
Yeah. Indeed, thank you for this. Yeah, for this nice summary. I think that's the whole message that came out of the study. And I also learned. I think it's important to highlight. And we had, I think when we started, we had a small team of maybe 5 core members designing the project. But we don't know everything, so it's so nice to collaborate with citizens and really take them serious, because so many people have knowledge from their own experiences, and they can be very valuable also for us as scientists when you design, especially projects related to the microbiome, because every lifestyle, every environmental factor could almost have an impact. So, and you need to find a way to make choices. And if it's only based on what's already in the literature, then you will just replicate what's already known. So you need to, it's nice to have enough novel sources of information for the projects like this.
Yeah, because you didn't just collaborate with citizen scientists. I love the way you were collaborating with, experts in nutrition and sociology and people who could communicate, and also marketing, very stylish, very fun, very humor-based as well. So there was, it's a very joyful project. But we're talking about the lactobacilli, and can you tell us a bit more about the variations? Because obviously if you're going from women age 18 to 98, what were you looking at? What are the other variations in terms of things like pregnancy or stress, or how did you, tell us more about that?
Yeah, very good point. So the whole idea was that we asked women to donate a sample so we could see their vaginal microbiome profile in a way as the community composition. But then also we had a questionnaire. It took women are almost 1 hour to fill in all these questions about lifestyle. And then we wanted to make associations to understand, okay, what, for example, when you get older, do you get less lactobacilli or not? And age was, for example, a key factor we asked. And then we wanted to rank how important they are. So is age the most important factor that determines your microbiome, or is it something else? And what turns out is age is important. So the older you get, the less lactobacilli you have. And that's a very important point of attention. It's linked to the menopause, because what's now well established is that you need estrogen to promote the glycogen production in your vaginal epithelial cells. And this glycogen is a sugar source. It's the food for your vaginal lactobacilli, because you don't have direct food in your vagina. So you need a source for nutrients for your lactobacilli. So if you have less estrogen, you will have less lactobacilli. But the picture was more nuanced. So the good thing is there are still many women above 60 years old already. They have passed their menopause, but they still have lactobacilli. So it's not at all bad. And there are women. So it's not only menopause. It's something. And menopause was not the most important factor determining vaginal microbiome. So it's something we want to explore further, because maybe in Belgium, we know a lot of women take hormone replacement therapy and so on. So we have to dig deeper a little bit. But so age has an impact, but it's not everything. Then the second on the list, the most important thing was having had children, and that's something we did not expect at all. So what we saw was, if a woman had a child, so a child given birth to a child, even if she had a miscarriage or an abortion, we saw less of Lactobacillus crispatus, so one species of Lactobacillus, but more of Lactobacillus gasseri, so another species of Lactobacillus. So it changed in species diversity. And we saw also more bifidobacteria, and they are important for the small babies, and also more Streptococcus. So it was not that it was a worse microbiome after pregnancy, but a different microbiome. And we don't understand that at all. So we have discussions now, what's happening during pregnancy? Is it because of the hormones, because of the anatomical changes? But because we also know already from other research, if you get pregnant again, then you have again more Lactobacillus crispatus. So it changed again. So there is a link between these key life stages. It's happening, but we cannot fully explain it at the moment.
If I could just ask you one thing there, Sarah, because we've talked on the podcast about the importance or the relevance of a mode of delivery. And again, we talk about essentially being sterile at birth, although I know there's some controversy about that. But if we assume that at the moment, as the baby comes through the birth canal, they collect these pioneers that then go to form the initial gut microbiome in the offspring. But we often talk about it coming from the mum's gut. Do you think that we're going to find the vaginal microbiome, the mother's microbiome, what she gifts her baby as she passes through the canal is going to be more important when you were talking about the bifidobacteria?
I think they're both important. But what we know from literature and a bit from our own research is that the vaginal microbes are also being transferred to the babies. There will be even part of the gut, but also on the skin. In the gut, they will not, like Lactobacillus crispatus, it's very much adapted to the vagina, but it's not so good. It cannot survive well in the gut. So it will rapidly be buried under all the other incoming microbes in the gut, but it will be there in the beginning. The bifidobacteria, they're also living in the vagina. They can also be transferred from the vagina of the mother to the guts of the baby and the skin of the baby, and they can survive in the gut. So I think our study was one of the first to show that bifidobacteria, they are dominant. They can be quite prevalent in the vagina. So I think now in future work, we want to really see whether the same, what we call strain, the same real bacterium is transferring from the vagina to the gut of a baby. For that, you need very good tools. And it would also mean the same. For example, we also found Lactobacilli reuteri, which are types of lactobacilli. They live in the vagina, not so dominant, but they're very good at surviving in the gut. And we also think that they will be transferred from the vagina to the gut of the baby. So it opens an all new area. But yeah, you're a GP, so during a delivery, of course, there's also stool being transferred and there's vaginal fluid being transferred. So it will be very difficult to dissect at all.
It is difficult to dissect. But again, if we're talking about narratives about women's body and women's health, I think it does change the narrative, because it's one of those things when you hear groups of women discussing their fears about giving birth, they're really embarrassed that they might pass some stool or they might, urinate and this completely changes again the narrative that actually this is a really important part of, your baby's gaining these important pioneers, these important bacteria. And it does change the story and perspective into a much more, positive one, and embracing, all parts of our body, even those that society have put a big taboo on. I think we need to kind of break that taboo and, embrace all this incredible information. So you're talking about age and pregnancy or parity as important sort of to the variations in the vaginal microbiome. What about periods? Did you ask people what part of the cycle they were on, or?
Yes, so I think that was the cycle was very important. You have the luteal phase and the follicular phase. So the phase where you have a lot of estrogen, you have also more lactobacilli. So that's crucial. And so it's also connected to the fact if women were on birth control pills with estrogen, they also had more lactobacillus crispatus. While if they were using a spiral or an intrauterine device with more progesterone, then they had lower levels of lactobacilli. So the hormones are, I think, next to age and having had children, were the third major driving factor of the community composition.
And what about, you did mention that most of it was on, obviously in Belgium, the European women. What do we know about racial or ethnic differences in the vaginal microbiome composition?
That's a very good question. I think it's one of the most, also in a way, how do I phrase it? So one thing, maybe first to say one thing because we work so closely together with sociologists and communication specialists, one thing we did not ask was women, what's your race or your ethnic group you belong to? Because it's like it's often connected to the way people live and where they come from. So we just asked them, where are you born? How long do you live in Belgium? And then we also asked about their lifestyle habits and what they eat and so on, because we thought, for the microbiome, it doesn't really matter what your skin color is. All these other factors matter. Because there are some very famous or the big microbiome papers who say that women from a black ethnic group, that they have a less beneficial microbiome, that they have less lactobacilli, more Gardnerella, and we think it's really biased because often they're from a lower socioeconomic status, they have less access to good food, less access to healthcare, and that also can all impact. So it's very crucial that this link to race and ethnicity should be carefully interpreted. So, and there are also, for example, there are studies that people, not only in Western countries, but also women living in Africa or in other remote areas, that they have a less beneficial microbiome, again, less lactobacilli, but also we don't know then how were they sampled. Often these studies are in sexual, or are with sexual sex workers, and then of course it's not the general population, or so the tendency or the view now is that in other countries and Western countries, women have a less beneficial microbiome, but I don't agree with that. I think it's just there are some mistakes being made in the sequencing. So that's one of the reasons we are also now starting a sisterhood project, and we ask women from different countries to launch a similar project and really targeting healthy women and sampling, and also to understand that indeed maybe they have a different microbiome with less lactobacilli, but that's still a beneficial microbiome, having the functions, keeping women alive, because there are many parts of the story that could still be beneficial. So we want to avoid the phrasing that women from other countries have a less beneficial microbiome. We don't know that.
I think it's a very, very important point you raise. And we know from the gut microbiome, you can't say good, bad, healthy, unhealthy, because we don't know. It's far too complex. It's like looking at a community in a city and asking if they're good or bad. It's just impossible to say. It's all to do with, not just who's there, but what are they doing? And but I think we need to be very sensitive about how we collect information, how we phrase it. And I suppose one of the things I really liked about your project was this idea that rather than people as subjects that are investigated, this was very collaborative and very empowering, and finding out about yourself and your body and your vagina and your community and contributing to knowledge seems to me a really exciting way to think about doing science in the future, empowering people to want to take part rather than taking samples and then judging them against some arbitrary scale. So it is interesting. It's also interesting to me because you can get these huge numbers. And I think if we look at one, I don't know if you call it citizen science or not, but it certainly started as that, the British Gut Project, which then morphed into Zoe. And this, people sort of argue, but they're getting huge numbers. So instead of I'm sure you'll be used to trying to get, as you said, 100 or 200 people in a trial, and you're actually having to shut down your trial because you got 6,000 registered in 10 days. I mean, it's really exciting because obviously the higher the numbers you have, that's going to iron out some of the issues with research. So what are the pros and cons of doing science like this? As you know, when you look at the project you did, can you give us 2 or 3 things that are for and against doing this sort of citizen science project?
I think you already mentioned one of the pros is that you have a lot higher numbers for your trial. Also, that it's in a way it's citizen reviewed, so your trial will be more societally relevant, and you know that you will ask the right questions. What I also liked about it is that we had a fun factor. We had also a nice design, and we had it was really a collaborative effort of the team. I learned a lot from it, so it's more rewarding as a scientist to do this trial because you have more a sense of belonging and that you're doing things that matter. And for example, we had a lot of postcards from participants who said, "Oh, we love your project. Keep on the good work." So we had people value what we do. So that was really nice. Cons, cons are, for example, it's much more expensive to do it. It takes time. I have a big group of researchers, and I think work-life balance is very important. But if you're doing a citizen science project, often we had to replan and we had to work a little bit later because we had to translate our website because suddenly we saw people from England were watching, and so we did a lot in addition to the normal working hours. So it's not something you can do every year, for example, and it will be a hectic time. And maybe a third major disadvantage is that some things are out of your control. So if, for example, we did sampling projects in the past with gynecologists, they take a vaginal swab from a dedicated place in the vagina. And they know where it comes from. They put the sample immediately in the correct vial. It goes to the freezer and everything is done properly. While now women took the samples themselves, we advised them how deep it had to be. We also said how many times they had to turn, and then put the swab again in the tube, and then put it in the fridge, and then send it to us. But it's out of our hands. We were not there, so we don't know whether they did it properly. Sometimes it took 1 day before it entered our lab. Sometimes it took 7 days. So there mistakes can have happened. But for us, it was counteracted by the fact that we had so many samples. So if one woman made a mistake, no problem. We had enough other samples. And then maybe also something very difficult was we wanted to study healthy women. And what's the definition of a healthy woman? So in the end, we said, if women say from themselves they feel healthy and they have a general good health, then for us it was okay because it's citizen science and we wanted to not make it a medical project. So there's no diagnosis, no GP who confirmed indeed you are healthy. Because sometimes it's okay, this woman says I'm healthy, but then we see our medical history and we say, oh, maybe it's not so healthy, but she was still allowed to participate because that was the idea of the project.
And we're talking about the ages that you covered from 18 to 98, and I understand that lactobacillus is dominant from puberty onwards. Do we know much about pre-puberty of the vaginal microbiome composition? I imagine that's quite a difficult area to examine and investigate.
Yeah, indeed, it's something I want to explore now in the future. So the dogma is again that it only starts during puberty, that you then get the increase in lactobacilli because of the estrogen. But there are some studies that it starts already a little bit earlier. And now I have my oldest daughter is 13. She's already menstruated, so it's also interesting to see. Okay, actually, we should try to do it in this new generation of girls. I think they would be willing to participate to a trial like that if we embed it properly. Maybe we are now also exploring. Maybe we don't need swabs, so we can discuss with them. Maybe sometimes the first urine can also be a good sample because it's linked to the vagina. We see the same type of microbes in there. And we want to also do a co-creational project with them. Would you be willing to participate in a project? What's important for you for this generation? Which questions do you have? Because they also need to make a lot of important choices, like which menstrual product are you going to use. You have now the cup, and you have this new type of underwear, and not only pads. So there's so many things that's happening, and it would also be a nice opportunity to break some taboos, and it's before they're becoming sexually active. So it would be nice to start a project not only to answer basic questions on science, but also help this generation with questions they have.
Did you ask about things like Tampax use, sanitary towels, vaginal douching, or was that over and above what you were studying in this project?
Definitely, we have asked about that. So one of the surprising things for me was that using a pad was negatively associated with Lactobacillus crispatus. So if we wore it the 24 hours before, then we saw less of the beneficial bacteria. And we saw the opposite for the cup. So we had a lot of younger women already using the menstrual cup, and they had more Lactobacillus when they wore it before. So tampons or Tampax, it didn't have a big impact. So maybe it's also good because there's also the stories about toxic shock syndrome. So we asked about pubic shaving. It had a little bit of impact, but not too much. We asked about douching. We had not so many women doing it. We saw a little bit. We saw a reduction, but not really a change in composition. We asked about sexual activity, for example, the way we did it was asking women to draw we had a heart shaped on the tube, and then they just needed to color it when they had sexual intercourse the last 24 hours. And we saw a big increase in diverse microbes coming in. But then after, if it was a few days later, then it returned to normal. So it's also part of it's a normal thing of the human body, and it's being tolerated.
But just being asked these questions is really empowering for women. Just being able to ask all these, that these intimate questions are of interest to someone and they're part of who you are. It's interesting, isn't it, that actually taking part in the projects, as you said, I can't get people to spend an hour doing a questionnaire, and yet they willingly gave up their time and were excited to be part of this.
Yeah, and that's a good point you raised, because we heard from many women that just thinking about the answers to these questions helped them with some issues there, or they were really reflecting. And some women, they told us that they were answering together, and then they were started to talk about it, and they it started conversations about certain taboo topics, which in the end were not so taboo. I think that was one of the main lessons I learned, that what I thought was going to be taboo was actually not so much of a taboo.
I suppose I would hypothesize that the project itself did that, destigmatized it, and changed it into something that this group of women owned and were proud to be part of. And that pride thing is So it's interesting how much that your project was not only collecting information, it was altering people's perspectives and society's perspectives, in Belgium, in that group of women, not, a large group of women. And that will go on, the discussion, they'll feel more comfortable talking, across generations. It really did open up a lot of conversations, I'm sure. And it's interesting that doing a science project like this in itself is therapeutic in some way that you probably can never measure. But you're getting the feedback. I mean, how many trials have you done where you get letters from the people in your research study thanking you for, having them in the research project, or, lovely feedback like that? I'm sure it doesn't happen. So if all this incredible information that you've got in mapping out the vaginal microbiome in this wonderful group of women who I thank wholeheartedly, and you truly are citizen scientists, and you're teaching me so much, and everything that you thought about wanting to belong to a project is coming to fruition, because we are all learning so much from your incredible contribution. So where does it, so if we're talking about, we've expanded our thoughts about the vaginal microbiome map, we're learning more about the effect of people's age, menopause, pregnancy, their periods. We didn't talk about stress, maybe we'll come back to that, but drugs, particularly HRT and the contraceptive pill, diet, again, we haven't spoken so much about that, but where do we move from saying, well, who is there, which bacteria, and we haven't even begun to talk about all the other microbes that will be there as well, to what are they doing and where are you going to take this project, or what can it help you with?
One thing we promised the women participants that in the future we would love to design novel diagnostics, and the other hand is novel therapeutics. So for the diagnostics, what we now have with this map is that we have a reference map of what a healthy microbiome more or less looks like, and then we use it for collaborations with clinicians on all kinds of disorders or diseases, and we compare. Actually, we basically compare, and we hope to maybe find novel diagnostics for certain diseases. And then the other part is we would love to explore certain novel therapeutic options. What if you don't have a beneficial microbiome? What if you lack lactobacilli? What can you do? Can we just transfer lactobacillus to this microbiome, and will it colonize, or does it need other friends? So how will it work? So because we did not only store the DNA of the microbiome, but we also really sort the microbes. The lactobacilli we have in our culture collection. Now we have thousands and thousands of lactobacilli from every woman, and we are doing microbiology screening experiments. Okay, which microbe is producing this novel vitamin or novel type of antibiotic, and we because we have so many samples, we can do it in a proper way. And then, so in the future, we also hope to develop living therapeutics.
So, Sarah, the one thing listeners will be wanting to know is about probiotics. I wonder if you could just introduce us to the idea of probiotics for the vaginal microbiome, and maybe start by saying what probiotics are.
So the official scientific definition is that probiotics are live microorganisms that, when administered in adequate amounts, confer a health benefit to the host. So it's a quite complex definition, and there are three key elements into it. It means that you have to take deliberately some live microbes in any kind of formulation, whether it's an oral formulation targeting the gut, but you can also use an oral formulation or oral product that even targets your vagina. And then you have to take a sufficient amount, so it should be really made with a purpose, and you need to have sufficient bacteria or live yeast. It's also possible. And then the third element, which is a key element, it should confer a health benefit, and that's very difficult to prove. It means that your bacteria should have, or the probiotics should have been tested in clinical trials, and that trial should have a positive outcome. And this trial could have been done in healthy subjects or people without any official complaint, or in a disease population. But studies in a disease population are very difficult to do with probiotics because they're supplements, and officially you cannot do a clinical trial for a drug with patients. So the trials are difficult to interpret, and the choices are difficult to make.
Wow, because when I knew we were going to be talking, I did look at the probiotics that are available in the UK, and it's like the Wild West. It's really complex, even as someone who has some understanding of how microbiomes work and the terminology. I found it really complex. So particularly for vaginal microbiome, some of the probiotics I looked at, they mentioned lots of lactobacillus, but none of the species which you've mentioned. So they seem to have a bunch of gut-related lactobacillus, but none of the crispatus or gasseri or jenseni that you mentioned. I mean, what's the landscape in Belgium, or how does it work?
I think the landscape is the same. I think there are numerous reasons why current probiotics don't have this vaginal typical species. One major reason is that the science is quite novel, and it takes on average 10 to 20 years before such products are being developed. But a second major reason is that the microbes, that species adapted to the vagina, like Lactobacillus crispatus, they don't grow well outside the vagina. So they're difficult to put into a product because they love that environment and the nutrients that are available, and they have difficulties in growing in these typical fermenters you need in industry to make these probiotics. So what industry is doing is they put other lactobacilli that are similar, and they put them into the products, which is not always ideal, but it will take time. But I believe there will be more women's health or vaginal probiotics on the market, but let's hope that they will be properly developed.
Yeah, it's really difficult for people because they just go in and see a big shelf of probiotics and maybe look at the vaginal microbiome probiotics. And it's complex because it's easier for me to say, oh, just steer away from them. And yet I know there's a lot of researchers doing really excellent work who are equally frustrated by the fact that anyone can, it seems, produce a probiotic that says it supports vaginal health. And some of them don't really tell you, they suggest 2 doses or 2 capsules a day forever. There's no duration. So it's quite expensive for people. And they could be taking something that's quite costly, that there's no scientific evidence of benefit. Would that be correct?
Yes, that's true. That would be correct. So that's also frustrating for us as scientists because regulations are currently so strict that producers or manufacturers making probiotics in a proper way, they are not allowed to put things on their packaging because they're not drugs, so they cannot make any claims. But the disadvantage is that other manufacturers that have less high standards, they can put the same on the package. So for consumers, the only possible solution is to go to the scientific literature, which is very complex, and then double check yourself because there are so many rules on how manufacturers can communicate on that. So we're also trying with scientists to find a way out of okay, where to set the barrier to make also distinction. Okay, sometimes there's good evidence out there, not fully ready to really fully recommend probiotics, but there are differences between different strains and different products and different species. So, and it can be of benefit. I think what I like to do as a scientist is look at the meta-analysis, so all the articles that are out there together. And for example, for when people are taking antibiotics, both for the gut but also for the vagina, that's really a condition where you can disrupt your microbiome, both in the gut but definitely also in the vagina. So then you're very vulnerable. For the vagina, definitely you can get yeast or fungal infections. And for me, that's scientifically definitely a case where probiotics could be of benefit. And you will also deplete the lactobacilli, so and it will be important to have to replenish them. And so then I would advise people to have, I don't know, I'm not allowed to say advice, but I think it would, if you want to explore it, it's really worth exploring it. And then preferably one with a lot of lactobacilli for vaginal health. What we are doing in our lab now in many different trials is trying to get a better understanding of which lactobacilli you take orally, which strains and species can really travel from the gut to the vagina via the natural routes, via the rectum and the area of the skin. You have some transfer because they need to end up in the vagina.
Sorry to stop you, Sarah. That's a really key point for me. I have to say, before I looked into this area, I presumed that if you were going to give a probiotic that was going to improve a vaginal disorder, it would, of course, had to be in a pessary or a vaginal form. And I find out that I'm wrong, and that you can get vaginal probiotics, but also people take it orally. Can you explain to me how can that be?
I think that's a very good point. I don't think you're wrong. You're absolutely right. It's just because of the law. So if you would put a lactobacillus in a vaginal, what I call formulation, a product that you can insert vaginally directly, it would be a drug. Then you really need to do different kinds of trials. And that's also something, for example, we would love to do now with our lab, but you need to run a trial like that. You need millions and millions of euros, so they're much more expensive to execute, and it will take time before these real drug products are being developed. So what the industry is doing is just making oral formulations, because then they can be studied but also put on the market as food supplements. But a big disadvantage, and you're 100% right, is that you only get a lower dose into the vagina of this lactobacilli. You can get to, and people have shown also in our lab, we see that there's transfer. So you get these strains end up in the vagina, but in lower dose. So is that really a problem? We don't know, because lactobacilli, they can grow well in the vagina. So maybe you don't need a lot and you can still have lactic acid production. And this lactic acid production will help you keep pathogens away. So mechanistically, this could work. But as you also feel, it doesn't seem ideal, but it might be suitable for the people who want to do something additional. That it can be of benefit, but as scientists and medical professionals, I think we would prefer the direct route of administration and also the drug route of clinical trials to explore which ones are the best to use.
It is intriguing, however, that even in reduced doses, that a probiotic live bacteria you take by mouth that travel down to the gut can end up in the vagina. Now, I presume that's possibly mainly because the anus and vaginal areas are near to each other, or would there be other pathways? Because we know, intriguingly, for example, in breast milk, that you can get transfer of microbes from the gut to the breast, which I find mind-blowing. Could there be such a mechanism here, or we don't know?
We don't know. I think it's a very good question. I think it could be because we know that dendritic cells or certain immune cells in the gut, they sample bacteria and they can cross across the body. And it's closely linked to it. I think it's possible, but it can also be through the skin. And it's something we would love to explore now in our ongoing research in more detail. But because it's happening, but in a low dose, it's very tricky to investigate. It's not easy.
So one of the conditions I see a lot of is bacterial vaginosis, which is a common vaginal disorder, which for anyone listening, it's traditionally treated with antibiotics, either in the vagina or more commonly in the NHS orally, metronidazole particularly, which, as you say, Sarah, causes tips the vaginal microbiome into an imbalanced situation, and there's a high recurrence rate. And I did see quite good effects from probiotics, which seemed in some aspects to be superior to antibiotics with favourable outcomes.
Yes.
Do you know much about that? Or
yeah, so I think the meta-analyses are quite positive and in favor of using probiotics. And for the moment, there doesn't seem to be a big difference between. So you need lactic acid bacteria or lactobacilli that produce lactic acid, and the good data that are out there are with strains that we know that survive well in the vagina. And I think that all lactobacilli have the property that they make lactic acid. So there seems to be some promise. And I know that many manufacturers are actually now using that, and often also similar strains. There are many women who have benefits from them, but also many others that don't. And that's also something we want to understand. Okay, when you give a new lactobacillus, when will it be able to have that function in your vagina? Does it matter which other microbes you have, or does your immune system has a role, or your hormonal status? And because you also need estrogen, because estrogen stimulates glycogen in the vagina, and the glycogen is the sugar that your vaginal lactobacilli needs to grow and to have sufficient numbers in.
So anyone who's listening here who thinks, gosh, this is very complex, you are absolutely right. This is so complex and I really struggle with it. And as I said, I have got a basic understanding. Now, most doctors don't. They're incredibly busy, super bright people, but they haven't got an interest in this area, which I have, and I'm struggling. Patients are told when they look at probiotics or when they look at websites or journals or magazines, go and speak to your doctor. Now, they are being sent to people who have no training in this area. It's something we need to be looking into. I would love to be able to admit my ignorance, but signpost people to a brilliant, easy to use website that they could, put in their disorder and their details and find the relevant studies. Is that something that anyone is working on?
Yes, I know there are some people working on it. I'm part of the ISAP, which is the International Scientific Association on Pro and Prebiotics, we are mainly focusing on the science, and we try to stay away from the recommendations, but others are doing that, and I have to look it up, and I can, if you want, I can send you some links. But people are trying to make these guidelines, which is very complicated because and we as scientists, we are bound to certain rules what we can communicate, and you have the manufacturers, they're bound, they have certain rules. They're not allowed to make any health claims, so they're very careful in informing the consumers, and they often only give information about their own product, and they don't compare products, which is what consumers want. And then you have the regulators who also are, especially in Europe, they're very strict on what can be communicated. But then on the other hand, you have so many products can come on the market without any claim, and then it makes it even more complicated. So there's definitely a need for educated people to inform the consumers on which choices which choices you can make, and what are the options, so that people can try, based on scientific evidence, they can try products for their conditions, knowing that not everything has been proven yet, and there's a lot of science that still needs to be done, but at least you have some options. Because I fully understand, if you suffer from problems, you cannot wait 10 years before the product will be on the market. And there are products on the market that, based on the current scientific evidence, could work. So it feels sometimes, it's difficult to also give the good advice.
It is difficult. And I suppose on this Microbiomedics podcast, Sheena and I try and be very pragmatic, because the guidelines for the NHS can take anything from 10 to 17 years to come. And people, if they've got a problem now, they want, evidence-based, best guess advice, a pragmatic approach. So anyway, we perhaps will do another podcast into this complex area. But going back to what Sheena and I do, which is a pragmatic approach using lifestyle medicine modalities. So that's looking at things like sleep, stress, physical activity, diet. Is there anything there that has a good evidence base that we know supports vaginal health?
Yes, actually, that's also what we have studied. And to our surprise, lifestyle had a rather big impact on the vaginal microbiome. And in general, eating healthy and sleeping well is also beneficial for vaginal microbiota. If you look at our own data on diet, and this has also been observed in other studies, we see that if you eat a lot of sugar, it's not beneficial for your vaginal microbiota. We see it's less associated with lactobacilli in the vagina. And the other way around, if you consume enough vegetables, and especially fiber-rich vegetables and nuts and seeds who are rich in phytoestrogens, then we also saw association with a more beneficial microbial.
Anything on fermented food, or not specifically for the vaginal microbiome?
Not specific. We saw some associations on fermented foods and probiotics. We also saw some more beneficial ones, but we're not sure yet whether it's a direct association or not. And it's definitely something we need to study in more detail in the future. But it could be, because fermented foods, they're naturally rich in lactic acid bacteria, often naturally rich in lactic acid. So it's biologically plausible that some of these microbes also have a beneficial impact on your vagina.
Okay, that's incredibly helpful. So those are positive take-home points that are going to be beneficial to your gut microbiome at the very least, and probably your vaginal microbiome too. And also I was thinking of things for people to avoid. So you've mentioned antibiotics are disruptors of the vaginal microbiome. I'm presuming douching and over self-cleansing could be in that category as well, to avoid.
Yes, definitely. Douching is a very bad idea. Using soap is actually almost the worst thing you can do, because soap has a high pH, while your vaginal microbes, they want a low pH. And so every time you douche, you would reduce the lactobacilli. So that's really not a good idea. Using antibiotics, of course, also not. But even also eating meat or sugar is then also, if related to lifestyle, is also not a good idea.
That's
not too much, at least.
Yeah, that's really, really good advice. So listen, I've taken so much of your time. This has been incredible, and there's so many questions I want to ask, but maybe we can have another podcast in many months' time and see where you're where you've got to in your research. How can listeners find out more about this project and future projects that you're doing?
So we have a website, isala.be/en for English, the English version. And there we really try to make a summary of all the research that is ongoing. But we also have guest people writing blog posts on everything related to vaginal microbiome and other aspects.
That's wonderful. And we'll put all those links in the podcast. So any listeners who are interested will be able to find them at the end. Sarah, thanks for your wonderful work. Long may it continue. And thank you to the citizen scientists, those women in Belgium. I hope they're listening to this podcast and can see how important their contribution is to this wonderful field of research. And finally, I think we should dedicate this podcast to Dr. Isala van Diest, who's the first doctor in Belgium only 100 years ago. And you've named the Citizen Science Project after her. So thank you so much. Thank you so much for listening to this episode of Microbiome Medics podcast. We really hope you enjoyed the content and we welcome your feedback. We'd love to hear any suggestions you might have for microbiome topics that you'd like us to cover. And we also appreciate listeners' questions and will endeavour to answer them in the next podcast.